Hello everyone, welcome in this video tutorial on modeling disorder in crystal structures about spatial position. The crystallographic program used are Olex2, Selexel and FragmentDB. All these programs are available free of charge for the academic users. The necessary files required for the refinement are used with permission from Dr. Peter Muller and from Mr. Sonk Edlung. The more detail about this particular example is available in this very nice book reference, Crystal Structure Refinement, a Crystallographer's Guide to Selexel. This is a very nice and informative book edited by Professor Peter Muller. So let's start with the tutorial part. For this, I am going to open the Olex2 and this is the molecule, an organic molecule which crystallizes in monoclinic space group C2 with two molecules in the asymmetric unit. The R factor is around 31%. The maximum peak is around 4.4 electron, and from here we are going to join the refinement. Up to now the refinement is very straightforward, and from here we are going to model the lattice solvent molecule. So from crystallization point of view, we are expecting some benzoic acid or some benzoate molecule in the crystal lattice. So for this, I am going to press this button compact and I am getting some benzoate here and one benzoate here. These two benzoate are looking very clearly in the crystal lattice. So okay, let's go to model these molecule first. For this, I am going to use FragmentDB. For this, press this button Tools and there is a tab FragmentDB. Press it and let's see for the benzoate. No, there is no benzoate available. This fragment benzoate is not available in the database, but no problem. For this, what I have to do, I will go back to work and I will rename them as a carbon and oxygen. Okay, so make these carbon, 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 carbon. Okay, okay, yes. And these should be oxygen. These should be oxygen. Okay, escape it. Let's name them systematically. Go to mode, naming, and suppose this is C1, C2, C3, C4, C5, C6, C7. This is O1, sorry. This is O1 and this is O2. Now type sort. It will sort all the atom in the .ins file. Go to edit and you will see the atom in this order. So these are the atoms of the benzoate: O1, O2, C1, C2, C3, C4, C5, C6 and C7. Okay, what I have to do next, I will refine. For the refinement program, I am using Selexel, 10 cycles and 30 cubics. Refine, and it will refine the atoms. And the R factor is dropped from 31% to 25%. And this is the benzoate molecule. Refine one more time, so the shift gets stabilized to almost 0. Yes, it's going to stabilize. Yes, now it's settled very nicely. Okay, so press this button compact and you will see another benzoate molecule here. Now I can use the coordinate of this benzoate molecule to model this benzoate molecule. How? Control Q and just select the all atom of this benzoate molecule. I have selected this benzoate molecule and I will go to Tools, FragmentDB, and here is a button Edit. Press this button and a new window will appear. Let's name this molecule as benzoate. And press this button Use selected atoms. Which selected atom? These selected atoms. Press this Use selected atoms and you will see these atoms will appear here. C1, C2, C3, C4, C5, C6, C7, O1 and O2. You can put some restraint like Rigu, C1 to O2, flat C1 to O2. So all atom from C1 to O2. And residue name is benzo, B-E-Z-O, benzo. And press this button Add as New. And you will see this fragment will be added into the list. Press this cross button and you will see the benzoate here. Benzoate written user. User means it is added by the user. So this is benzoate. Now what I have to do, I have to press again Control Q and the Q picks will appear here. Let's adjust the size of Q picks. First tell 20, so the thermal are looking nice now, and then press zoom 300. Okay, so now the Q picks are looking very nice. Now press this button and you can see the benzoate will appearing here. Okay, so now what is the procedure? This benzoate is on the journal position, so I am using part 0, free variable 1 and occupancy is 1. Press this button Fit and this benzoate is appearing here. Now match 3 atoms. First this carbon with this carbon, this carbon with this carbon. Okay, the two matching is enough. Yes, it's fitting very nicely. Now what I have to do, press this escape and refine. So now you can see the R factor is dropping to 20% from 25%, and this is fitted very nicely. And refine one more time so the ship gets stabilized. One more time. Good. So now there are two benzoate molecule, one and second is here. Refine one more time. Good. Now if I press this button compact, I am getting some electron density here with Q peak intensity 2.97 3.44, 4.22, and same electron density is appearing this side also. So what this electron density indicates? Okay, press Ctrl G, grow, and you will find it's growing and it's also growing. So if you see, this electron density is forming some strange molecule like some benzene ring here, and some electron density are appearing here. If I ignore these two electron density, then it looks like a benzoate, but it's appearing something like benzodione type moiety, very strange molecule which is not looking like a benzoate molecule, because this is a disordered benzoate about the crystallographic two-fold axis. That's why it's looking very strange like this. So this is actually the benzoate molecule tilted around the crystallographic two-fold axis. Okay, let's understand it by drawing in another program, or drawing in through a diagram or C2 axis separately. Here you can see, yes, this one. You can see the benzoate molecule is here. If you ignore the hydrogen on the oxygen, then the benzoate has a C2V symmetry. But in this case, the C2 axis is not passing through the middle of the molecule, but it is passing like this. So 1, 2, 3, 4, 5, and 6. These 6 atoms are appearing in the asymmetric unit, and the remaining atoms are donated by this C2 axis from these atoms. So the situation is not like this in the present case, but the situation is like this. So overall we are getting a situation like this. So the molecule is actually tilted about the C2 axis, and we are getting this type of electron density. This type of structure we are getting from the growing the cubics. So this is actually situation. This green oxygen is overlapping on the blue oxygen, and we are getting a strange cubics or strange structure from the cubics by growing command. Let's come back to the Olex2, and we are getting this type of strange structure. This is benzene, and if we ignore these two electron density, this is looking like a benzoate. So let's model this benzoate molecule using the FragmentDB. Okay, so what I have to do, I am again going to the benzoate, and here I am using part -1, because the disorder is about the crystallographic two-fold axis. So we need no two part 1 and part 2. We need only one part -1, and occupancy is only half because the disorder is about the two-fold axis. So now what I have to do, I have pressed this button fit, and I have to fit the molecule. So this is the carbon, this one here, this carbon is here, and if I just rotate, I will get a nice fit. Yes, almost a very nice fit. Now press escape and let's refine. Okay, so R factor is further down to 13.3%, and you can see this molecule here. Yes, you can, if you can see carefully, this molecule is like this one, like this one. This is the present situation. So if I press fuse, okay, so this is the fitted molecule with half occupancy about the, and this is disordered about the crystallographic two-fold axis. So this is modeled very nicely. I am left with one more molecule. Press this button and you can see the another molecule here. This is the carbon. Carbon, these are two oxygen, and the remaining carbon will be appear here. So let's first fit it. I am again going to fit the part -1. Occupancy is half because the second molecule is also half occupied and it is disordered about the two-fold axis. Press this button fit, and this carbon should be here, this carbon should be here, and if I tilt it like this, I will get a very nice fit. Yes, press escape to work and refine. So now you can see the R factor is further down to 7.55%. Now the maximum Q peaks is only around 0.7 electron. So we have modeled all the electron density in the lattice. Now next step is anisotropic refinement, type anis and refine. So R factor is further down to 5.30. For clarity, I am just going with the lines 20. Yes, that's good. Now next step is hydrogen add, H add. Press this button add H, and it will add the hydrogen on the lattice solvent benzoate and benzoic acid. Now R factor is further down to 4.59%. That's nice. Okay, assume. Okay, now the electron density is around 0.3 electron. So almost everything is modeled. Now we have modeled 4 molecules in the lattice, out of which 2 are fully occupied and 2 are half occupied. So there are only total 3 molecules. 1, 1, half, half, 3. So how many molecule per molecule in the asymmetric unit? So I am just selecting these hydrogen, delete, and again hydrogen add. So now it's looking nice. Okay. Yes. So this is actually nitrogen. It has NS3. So it is NS3 plus. It is NS3 plus. So there is overall 2 plus charge on the main molecule. So we need 2 minus. Okay. So we need 2 benzoate. So the 2 molecules in the lattice which are fully occupied are benzoate, and which are half occupied, they are benzoic acid. So I have to put hydrogen on these 2 half molecules. Here this is fully occupied, this is fully occupied. So these are 2 benzoate and the remaining 2 are benzoic acid. Okay, so this is part -1. So there should be hydrogen, half hydrogen, and another molecule is here. This is in part -1. Yes, so 1 and half, half hydrogen should be there. Half hydrogen is there and half is there. For this, put the hydrogen, hydroxy hydrogen, go to tools, and here hydrogen atoms, press this button and select this hydrogen and this hydrogen. It will put the hydrogen on these two oxygen atoms. Press escape and go to refine. And now you can see the hydrogen are on these two oxygen atoms. Now press this button to update the molecular formula and refine one more time. So R factor is now 4.27%. Refine one more time so the shift gets stabilized and the weighing scheme become green. It will require some more cycle to convert the refinement, and you will see the now shift is almost settling. The goodness to fit is now around 1. Completeness is 100%. The shift is getting further down. There is no need of the EXTI. Now it will become green. This is also settling down slowly. Now you can see everything is almost looking green. Refine. Almost the refinement is converged. It will need some 1 or 2 more refinement and it will settle down almost. Yes, it's almost settled down. F observed versus F calculated graph is now looking very nice and everything is settled down very nicely. If you have any question regarding the particular tutorial, you are welcome. Thanks for watching the video. Okay, one more thing I want to add. If you visualize the molecule using the program like Mercury, then you can see the disorder very nicely in the benzoate. For this, just I am deselecting the remaining molecule. Okay, just hide these atoms and you can see the molecule. So these two are the fully occupied benzoate and these are the half occupied. If you see the asymmetric unit, it is looking like this. If I go into packing, then you can see this benzoate is only half occupied and it is along the two-fold axis, only half in this unit cell and half in the neighboring unit cell. Okay, thanks for watching the video.